Hello, my name is Joanna Lemley. I'm a wetland ecologist with the Colorado Natural Heritage Program at Colorado State University. The following presentation will include key concepts in the Field Protocol for Lentic, Riparian, and Wetland Systems, the official protocol of the Riparian and Wetland AIM program. You can follow along in sections 1 and 2 of the protocol. Riparian and wetland areas are transitional between terrestrial or upland environments and aquatic environments like lakes, rivers, and streams. They have high ecological value because they cover a small fraction of the landscape, only 1 to 2% in western lands outside of Alaska, yet they support high biodiversity and provide numerous other ecological services. Riparian and wetland areas help maintain clean and abundant water within watersheds. They store soil carbon, provide recreational opportunities, have cultural and historical value, and support livestock production. Given their importance in the western landscape, there is a need for consistent monitoring of riparian and wetland resources across all public lands. While BLM lands are generally known as sagebrush country, these lands contain a surprising diversity of riparian and wetland resources. Each of these photos represents a wetland on BLM public lands. Where there is water in a dry landscape, we all flock to it. That includes recreationalists, wildlife, and livestock. To address the need for a riparian and wetland monitoring program, the Field Protocol for Lentic, Riparian, and Wetland Systems was developed through an interdisciplinary collaboration between the BLM National Operations Center, the Colorado Natural Heritage Program at Colorado State University, and the U.S. Forest Service. The protocol draws on several existing protocols developed by the BLM and outside agencies, including the terrestrial and lotic AIM protocols, BLM's Multiple Indicator Monitoring, or MIM, and EPA's National Wetland Condition Assessment Protocol. This is the official protocol for the BLM Riparian and Wetland AIM program, and it joins the family of AIM technical resources. The goal of the Riparian and Wetland AIM program and the Field Protocol for Lentic, Riparian, and Wetland Systems is to provide a standard way to monitor wetland and riparian resources on public lands in order to inform policy, planning, and state and federal regulations. Data collected using this protocol can be used to evaluate the effectiveness of land management actions on maintaining or improving the biological, physical, and chemical integrity of riparian and wetland resources. The data can be used to determine the condition, track trends, and measure annual use of riparian and wetland systems. The protocol is applicable at multiple spatial scales, from fine scale, including site-specific, pasture, or allotment scales, to broad scale, such as ecoregional, state, or national scales. The protocol and intended uses follow the principles outlined in the BLM AIM strategy and the BLM National Aquatic Resources Framework, both documents shown here on this slide. Resource monitoring is an essential component of adaptive management. Monitoring begins with identifying clear management goals and questions, which in turn lead to monitoring objectives that specify when, where, and how monitoring data are collected. The process of selecting sample sites is called a sampling approach. The points selected for a given project are called its sample design. There are two primary sample approaches or sample designs. First is targeted monitoring. In targeted monitoring, sites are hand-selected to address a specific monitoring objective, often due to management concern or recent or upcoming management action. Targeted monitoring is appropriate for site-specific evaluations of treatment effectiveness or specific areas of concern, and can be used to document a reference condition, establish repeat monitoring areas, monitor known habitat of a rare plant or animal species, or track changes that result from management actions like grazing or restoration. Data from targeted sites can then be used to compare one individual plot against another or to calculate trends over time within the same plot, but cannot be extrapolated beyond the specific plot. In random sampling, sites are randomly selected from all wetland and riparian areas within a specific geographic area. Every member of the target population has a known chance of being selected. Results from random sample designs can be extrapolated to provide statistical valid estimate of condition and trends across an entire population or study area with known levels of precision and accuracy. The Field Protocol for Lentic, Riparian, and Wetland Systems is intended for vegetated riparian and wetland areas, which are sometimes referred to as lentic areas. These areas include wet and mesic meadows, marshes, seeps and springs, peatlands, which are bogs, fens, and muskegs, vegetated playas, vernal pools, prairie potholes, kettle ponds, vegetated drainage ways, swales, riparian shrublands and forests, oxbows, beaver complexes, floodplains, and the margins of ponds, lakes, and reservoirs. There are three critical concepts included in the definition of applicable ecosystems. That is riparian areas, wetlands, and lentic areas. Those will be explored further in a separate presentation. This slide illustrates some of the more common types of riparian and wetland areas sampled with the protocol. The two types at the lower right can also be described as floodplains. To further bound the environments appropriate for sampling, the protocol contains a set of 6 field criteria that must be met. To be sampleable with this protocol, a site must have perennial vegetation, at least 10% cover of perennial vegetation under typical growing season conditions without disturbance, meaning heavy livestock use, wildfire, flooding. Hydrophytic vegetation. With an existing canopy cover, the site must be dominated by hydrophytic vegetation or species rated as obligate, facultative, wetland, or facultative. Hydrology. The site must have evidence of hydrology influenced by surface or groundwater at some point in the growing season. And the majority of the monitoring plot must be beyond the immediate banks of an unvegetated, active river or stream channel. No more than 10% of the monitoring plot can contain an unvegetated active channel. Shallow water. If standing water is present, the site must have no more than 10% cover of permanent standing water deeper than 50 centimeters or 20 inches during the growing season. Area for transects. The site must have sufficient area to accommodate 3 25-meter transects with individual transects spaced at least 5 meters apart. And minimum width. Narrow sites must have a minimum average width of 2 meters. Methods within the protocol are divided into covariate, core, contingent, and annual use methods, as listed in Table 2 of the protocol. Covariates are methods that characterize sites for classification, stratification, or to determine the potential natural conditions. Core methods are relevant across many different ecosystems and have widespread cross-program applicability. They are required for all monitoring. Contingent methods have the same cross-program applicability, but are only measured where applicable to specific management objectives. Annual use methods are included for monitoring the degree of vegetation use or soil alteration related to grazing or browsing by livestock, wild ungulates, wild horses and burros, or human activity. Covariates help describe the site being sampled and include photo points and basic descriptive information like elevation, slope, and aspect. And they are all collected following the terrestrial AIM methods. Classification, hydrology, and soil profile descriptions are all specific to riparian and wetland environments and follow methods outlined in EPA's National Wetland Condition Assessment. Lastly, data on natural and human disturbances are collected. These specific methods have been adapted from multiple sources, including EPA and other AIM programs and NatureServe. There are 4 core methods in the protocol. They include species inventory, line point intercept for vegetation cover and ground surface attributes, and vegetation height, litter, and water depth. These 3 methods were adapted from terrestrial AIM methods. The 4th core method is woody structure, which was adapted from 2 separate methods in the multiple indicator monitoring or MIM protocol, those being woody species height and riparian woody species age class. Contingent methods in the protocol include hummocks and water quality. The hummocks method was newly developed for this protocol, and the water quality methods were adapted from LODIC AIM. Annual use methods include soil alteration, stubble height, and riparian woody species use, which were adapted from the multiple indicator monitoring or MIM protocol and older BLM technical references on annual use monitoring. The final few slides in this presentation cover elements of Section 2 of the protocol, titled How to Use this Protocol. The first element is the standard monitoring plot. Data for all methods and covariates are collected within a defined area surrounding or near a specific sample location, which is a set of spatial coordinates selected in advance using the appropriate sample design, as explained earlier. The sample location, also sometimes called a sample point, should be located in or near a riparian wetland site. The defined sampling area is called the monitoring plot. Data collected with this protocol represents conditions within the monitoring plot, and not necessarily across the entire riparian or wetland site. For small riparian or wetland sites, the plot may encompass the entire site. For large riparian or wetland sites, the plot will only represent a portion of the site. For site-scale targeted monitoring, plots are often strategically located in areas where management action may have the greatest effect. If the targeted site is large and complex, more than one monitoring plot may be needed to adequately represent conditions. For population-scale random sampling, monitoring plots distributed across the landscape will represent the range of conditions within all riparian and wetland areas across the landscape. The standard monitoring plot configuration is the spoke layout. The spoke layout is a 30-meter radius circle that encompasses approximately 0.3 hectares or 0.7 acres in area. The spoke layout is often but not always centered on the sample location. The spoke layout includes 3 25-meter transects spaced 120 degrees apart. Each transect starts 5 meters from the plot center. The spoke layout should be used whenever the site can accommodate a 30-meter radius circle. For sites that are too small or narrow to accommodate a spoke layout, the protocol contains 4 alternate layouts in section 4.0. Those are transverse layout, diagonal layout, linear layout, and a mixed layout that incorporates elements of multiple layouts. The figures on this slide illustrates the 4 primary layouts. The 5th layout, called the mixed layout, incorporates elements of multiple layouts and is used in sites with variable dimensions. Each layouts share one fundamental characteristic: 3 25-meter transects spaced at least 5 meters apart and evenly spaced across the monitoring plot. Each method within the protocol can be carried out independently. In practice, a suite of methods is carried out during the same sample visit. Appendix C in the protocol includes time estimates for each method. Figure 3, included here, illustrates the recommended order of completing all methods. The recommended order assumes a 3-person crew, with 1 crew member leading the vegetation data collection, another crew member leading the soil and water data collection, and a 3rd member supporting the others. For some crews, these roles will remain consistent throughout the field season, but other crews may want to change roles at different sites to build skills in all components of the protocol. I will not go through this flowchart in detail because it is hard to understand how the pieces fit together before learning each method, but it is important to see that there is a recommended flow that maximizes efficient data collection at each site. While this flow is recommended, site conditions and monitoring objectives will dictate which methods are used and the actual order of sampling. For instance, not all sites have surface water, woody vegetation, or hummocks. Given the diversity of vegetation of riparian and wetland systems, sampling will be different at each site visited. Lastly, I will cover the timing of data collection. The optimal time to sample core methods is when vegetation is most identifiable, annual soil disturbance is minimal, and water levels have receded. In the western United States, the optimal time is typically late spring through summer. In contrast, the optimal time to sample annual youth methods is during the use period or immediately following livestock grazing or other activities. Practitioners should evaluate the purpose and use of the data to determine when to sample. In some cases, more than one sample visit may be necessary. This concludes the introduction to riparian and wetland AIM. Thank you for listening.